Good afternoon, and welcome to the 2021 Invent Penn State Venture and IP Conference Virtual Tech Showcase. My name is Harl Tolbert. I'm Assistant Vice President for Research, and I direct the Office of Technology Management here at Penn State, and I will be serving as today's Tech Tournament MC. I'd like to thank my colleagues, Dr. James Delatre. James is our Associate Vice President for Research, and he's Director of the Office of Entrepreneurship and Commercialization here at Penn State. And I'd also like to thank Jim Pietropalo, who is Associate Director for the Penn State Office of Entrepreneurship and Commercialization and an active member of the Invent Penn State team. Dr. Delatre and Jim Pietropalo mentored all of today's companies, and we're thankful for their hard work and dedication. I would also be remiss if I did not thank our incredible roster of 22 sponsors and for their continued support. Without them, the virtual and in-person Penn State Venture and IP conferences would not happen. Thank you. You can find our logos prominently displayed during our virtual event over the next 2 days. You can also find information in our program book and on our website. Thank you again. Today, 8 companies representing the best and brightest of Penn State's early stage innovators will go to head-to-head in competition. You can read about each one of the companies in your Tech Tournament guide. Each company will make a 3-minute presentation, which will be followed by 5 minutes of Q&A and follow-up from our panel of judges. Cash prizes will be given for first, second, and third place winners. The third place winner will receive $25,000. Our second place winner will receive $50,000, and the grand prize winner will be awarded $75,000. We're thankful to Penn State University for their generous prize money and continued support of this program. The winners of this competition will be selected by our panel of judges and will be announced tomorrow directly following our keynote speaker, Kenneth Fraser, Chairman of the Board and CEO of Merck & Company, Incorporated. Kenneth will begin speaking at 2 o'clock. I would like to now introduce today's distinguished moderator and panel of judges. Today's moderator will be Erica Swift, Associate Director of Penn State's College of Medicine Center for Medical Innovation. And our judges will be Dr. Alice Chang, Associate at Upfront, Ilana Diamond, Managing Partner at 412 Venture Fund, and Bruce Shook, President and CEO of Vesper Medical Incorporated. You can read more about today's judges and moderator in your Tech Tournament guide. I would now like to turn the program over to my esteemed colleague, Erica Swift.
As Harl said, my name is Erica Swift, and I am the Associate Director of the Center for Medical Innovation at Penn State College of Medicine. I will be serving as today's Tech Tournament moderator. A few notes about today's program before we get started. As Harl indicated, we will have a total of 8 presentations today. Each presenter will have 3 minutes for their presentation and 5 minutes of Q&A from the judges. You can find information about each presenter in the Tech Tournament booklet link that was sent to you. Now, let's get started, and good luck to all the presenters. Our first presenter is Joe Protruvo of Bio-Based Extraction of Rare Earth Elements. Joe, you may begin.
Okay. All right. So I'm Joey Protruvo. I'm an assistant professor of chemistry at Penn State and inventor of a bio-inspired method to extract rare earth elements. Rare earths are a group of metals that have irreplaceable functions in technologies like cell phones, electric cars, fighter jets, and much more. The demand for rare earth components of these products increases every year, as you can see down here. But the US currently relies on China for most of its rare earths. And as a result, the Department of Energy has labeled rare earths as critical materials to US technology and national security. Sorry, let me hide the thumbnail here. Okay, so the problem is not so much that we don't have domestic resources of rare earths. We actually have vast resources, but they tend to be of relatively low grade. So current technologies that to harvest these metals use toxic chemicals, and they don't work well on low concentration sources. And as a result, we throw away billions of dollars worth of rare earths every year. Our solution to this problem is a natural protein that we discovered that has evolved over millennia to bind rare earths, and only rare earths, very tightly. Its combination of selectivity and affinity for rare earths is unmatched by traditional methods. It's robust and it's reusable, and it therefore it's able to make otherwise inaccessible low-grade sources of rare earths accessible. Our technology can be implemented in a number of ways, but one example is shown here. So acid mine drainage, or AMD, is produced both by operating mines and abandoned mines, and it's a big problem in Pennsylvania and in Appalachia. This drainage contains many different metals, including rare earths, and as a result, it has to be treated before water ends up in streams and rivers. And so what we conceive is that an immobilized form of our protein, such as on a column, can be integrated into an existing AMD treatment facility to recover the rare earths with high purity in a single step for further processing. We have 2 patents that are pending on our technology, and we're currently working with a mining company on further process optimization and development, as well as economics. My student and I discovered this protein, and we're the experts on how it works. And we've teamed up with researchers at Lawrence Livermore National Lab who are experts in development of biotechnologies for metal extraction. We have funding from the DOE, from Penn State, and we've participated in the I-Core Site program, which has helped us to begin to learn from the market about how we can meet our needs, and we're continuing those efforts to further refine our technology. Thank you.
Thank you, Joe. We will now start the Q&A session, beginning with Alana. Alana?
Hi, Joey.
Hi.
That was a really interesting presentation.
Thank you.
You mentioned $5 billion of discarded rare earth materials, and I'm wondering how your process affects the cost to deliver the end product, if you know that, and also I'm trying to get a sense of overall how much this could affect the market. 5 billion, what percent is that? What percent can you add to the total market of what we need? So do we need 100 billion, or do we need 10 billion and you're providing half of it?
Yeah, that's a good question. So it's hard to answer that question. I can say that what the estimate is that there's millions of kilograms of rare earths that are produced in acid mine drainage in just Pennsylvania and West Virginia alone every year. And so that's about 1% of the total world market of rare earths. So, these sources are somewhat localized to mines, but each mine would, you would have to implement this at each location in particular. If that, I know there are not dollar amounts in that answer, but.
Well, that's still, I think 1%, that's a significant amount of the world's, the world's supply. And how does your process affect the cost to extract it? Do you have any idea about that? So the end product cost, how will using your process versus mining it in China, let's say?
Right. So that's going to depend on, so we're still in the process of optimizing the economics of our process, right? We've been operating at laboratory scale. We would need to scale up to, and obviously the costs don't work at laboratory scale. But if you're able to produce the protein in a fermentation facility, do everything at scale, then I think we can begin to be competitive based on the costs that I've looked up for, protein production and conjugation. I think we can begin to be competitive with those locations. And one thing that I want to add is that acid mine drainage is relatively enriched in the heavier rare earths, which are much more valuable than the lighter rare earths. So the costs of those depends on the specific element, but it's like $100 to $1,000 per kilogram. And so if those are the prices, then given how much protein production costs, I've seen numbers like $60, $70 per kilogram, but we can reuse the protein many times. And I think at the end, what it comes down to is how many times will we be able to reuse the protein.
Thank you.
Yeah.
I believe I'm next. Joe, congratulations. Very interesting idea, solving a very important problem. A few quick questions. One is, who is the ultimate customer for your technology?
Yeah, I think probably who we would be selling to would be, or who would be working with would be the companies that are involved in management of the wastes from these mines. So from what we found so far, it seems that the mines will partner with particular companies that manage these treatment facilities. And so I think it would probably, what we probably would end up doing is partnering with those companies rather than with the mines themselves, although we're still learning more about the market.
And in terms of from a business model perspective, do you envision just sort of selling the hardware, once and done, or do you envision somehow sharing in the, the value created by selling the rare earth metals? What are your thoughts on business model?
Yeah, so to be honest, it's the rare earths area is somewhat of a mafia, and it's been a little bit difficult to learn about all of these details. But what I envision at this point is that we would be selling a product to the companies that would be managing these wastes. I'm not exactly certain how the money or the value that's produced from the rare earths, how that would be distributed among those various partners, but that's the best that I can answer at this moment.
Thank you. I'll turn it over to Alice.
Thanks, Joey. So you mentioned a couple times that the proteins were reusable. Can you just talk a little bit more about the tech behind that? Like, do you have to repurify them after you use them, or how does that process work?
So even though the protein binds rare earths very tightly, it's also very easy to remove the rare earths, to desorb them. So you just would have to do a desorption step, and then you would be able to load the protein again with rare earths. And so we've tested in the lab, we've tested, we've run samples through the same protein many times and have not seen disintegration of the protein. So the expectation is that we'll be able to use this, dozens and dozens of times in the field.
Okay, and quickly, how much more R&D do you think you'll need to do before you're ready to deploy this product?
Yeah, I think one of the biggest things that one of the biggest questions we need to resolve is the matter of scaling up production of the protein. We've done a lot of investigation into the actual performance, and those studies have gone very well. I think the major issue is the fermentation of the protein, which there are methods to do this. We just need to implement them with our protein.
Thanks.
you. And we are now at time. Thank you, judges, for your questions. And thank you, Joe, for your presentation. Very nicely done.
Great.
Our second presenter today is Michelle Washko of Respona Therapeutics. Michelle?
Thank you, Erica. My name is Michelle Washko, and I'm representing Respona Therapeutics. Respona is developing novel therapeutics for respiratory conditions. The problem that we're attempting to address is influenza-related morbidity, mortality, and negative economic impact. In the US, the direct economic burden of influenza exceeds 10 billion per year, and the combined direct and indirect costs exceed 87 billion per year. The strong human toll can be found in the table at the right. The opportunity we have is to introduce a much needed addition to the therapeutic arsenal. Respona is developing a monoclonal antibody for complications arising from respiratory infection. The initial target is hospitalized influenza patients at high risk. We would then proceed to adults of average risk. The secondary target is patients in ambulatory settings. The total addressable market for these two segments in the US alone is estimated at between $150 and $350 million per year. We see additional opportunities in prevention and emerging coronaviruses. Collectively, the combined opportunity exceeds 2 billion. The technological foundation is a monoclonal antibody specific to surfactant protein A receptor SPR210. This is not pathogen specific, so not susceptible to resistance due to pathogen mutation. We target the host, that is the patient, rather than the virus. We address the maladaptive autoinflammatory response, but importantly, we do not target an individual cytokine pathway. In short, the mAb recalibrates the immune response from inflammatory to reparative. Proof of concept studies show that influenza A infected mice treated with Respona's mAb recover more quickly, have better post-infection lung function, and show better lung histopathology than untreated mice. The competition. There are two antivirals indicated for treatment of influenza. Baloxavir and M2 inhibitors are also used, but infrequently in the US. Antibiotics are typically reserved for special use cases. There are several mAbs either in development or on the marketplace, but none of the approved mAbs are indicated for flu. And importantly, again, Respona's antibody does not target individual cytokine pathways like these. We have a very strong team, both on the scientific side and on the management side, and we are currently in negotiations to onboard our first 2 scientific advisory board members. Here you see the commercialization plan laid out for you quarter by quarter, culminating in an IND filing anticipated in Q1 of 2023. If we were to be awarded the funds, we would use it for comprehensive analysis of the lead humanized monoclonal variants and for robust IP prosecution in both Europe and Australia. In short, the case for Respona is a strong one. We have a compelling value proposition for both patients and the healthcare ecosystem. We have a unique target with highly promising preclinical data. The IP position is strong. The scientific and management teams are both also very strong, and we have early external validation. And with that, I'll leave it to questions. Thank you.
Thank you, Michelle. Nice presentation. Okay, so we will begin the Q&A, starting with Alana.
Hi, Michelle, that was very nice. Thank you, and very interesting. You talked about a large market size there. And when you're thinking about your total addressable market of 150 to 350 million, I'm curious, what % of that total addressable market is actually your specific solution? Can you actually address sort of your service addressable market? And how did you get to those numbers?
So those data were driven largely by a paper that laid out the costs. Actually, if we go back to this, can I go back to my slides and show you the second slide there? That might be helpful. I'll share that. So if you look at the chart here on the right, this is data that comes from, this is sort of a compilation of data from a variety of sources, primarily in that particular chart, the CDC, but there's a lot of literature on influenza out there. And we've looked at some papers that look at the total economic burden of influenza, particularly one, I can't quite remember the author's name, but it's cited in the one pager that's part of the packet. And that particular paper looks at average influenza costs over a 10-year period. And what we did is look at the costs for both inpatient and outpatient folks, and we looked at the total economic burden on average, and we looked at, we combined that, we triangulated with the data for the antivirals that I referenced and what their market share is and what they're bringing in annual sales, and that's how we arrived at 150 to 350 million.
Thank you.
Sure.
Okay, Bruce, do you have any questions?
Yes, Michelle, very nice presentation. Thank you. A few quick things. One is, have you had any preliminary discussions with FDA about, clinical trial requirements in your regulatory pathway?
We have not gotten there yet, no. We have been consulting with various experts in the field. There were a bunch of different pathways that we could take here. We do think, based on these early discussions, that we would start with inpatient folks, and then beginning with high-risk adults, those over 55, those who already have underlying conditions. But there are certainly other methodologies, and for that, we are looking to consult with additional clinicians. We do have Dr. Haberlak, who is a Penn State person, Penn State faculty and clinician, who was advising us, and also the SAB members who would be coming on board also have particular expertise in that area. So the short answer to your question is no, we've not had preliminary discussions with FDA. We know there are lots of options here, and we know that we need to do additional research with folks who are actively in the clinic to understand exactly what our opportunities are there and what the appropriate pathway would be.
Okay. And how much cash do you estimate it'll take you to get to your IND filing, what you outlined in your Gantt chart?
To get to the IND, we think that probably we need about, I would say, between $1.5 million and $2 million, probably get to that IND filing. A lot of that would be in the cost, right? As I think many of you and around the room today would understand, is that to have the material made in the quantity that's sufficient for a clinical trial is going to be the biggest lift there. That could be as much as $1 million. We don't really have a visibility on that yet. It could be appreciably less, but we don't know that yet.
And are you confident it is manufacturable at some reasonable scale?
There's no reason to think that it won't be. The early affinity data looks good. So we feel like it's probably not going to be huge amounts of material. But again, that remains to be seen until we consult more with drug formulation folks and explore those into further detail with CROs. We have been in discussions with CROs who have reputations in this space, but I would be exaggerating if I said we have strong visibility on that just yet.
Thank you.
Mm-hmm.
Hi, Michelle, thanks for that presentation. I have a question about the discovery process. So how did you originally come up with this indication? Was it you already had this indication and you were screening for it, or did you discover the antibody first and then try and look for an indication later?
So I'll do my best to answer that accurately, but that's a question that's perhaps best reserved for our scientific founders, Drs. Kronios and Christensen. My understanding is that he identified the target first, the surfactant protein A. That is his discovery, that SPR210. And his early work was all with influenza. So what I haven't stressed here in this presentation is that it looks like we have opportunities in lots of areas of the respiratory space, but all the early work has been done with influenza. There is some, it's mostly viral, some bacterial influenza as well.
So you were hinting at potential other indications.
Yes, so there's every reason to believe that we could attack some other coronaviruses for sure. Prevention, again, theoretically, looks like it should be entirely plausible. We also think because of the reparative nature of the MAB, we believe that we might even have applications in fibrotic diseases. And actually, we've explored that in conversations with some potential SAB members with whom we've had discussions. And that seems to be of great interest to those folks as well.
All right, thanks. That's it for me.
Yeah, great. Thank you, judges, for your questions. Michelle, great presentation. Thank you. Our third presenter is Joe Sassani of Elkos RX. Joe, you may begin.
Hello, I'm Joe Sassani from Elkos RX. Thank you for the opportunity to introduce topical naltrexone that will be changing the standard of care for early stage diabetic wound healing. Our team is broadly based with representatives of pharma, basic science, and medicine. Since we last presented at this forum, we've developed new naltrexone GMP formulations that are safe and even more effective in speeding the healing of wound care in diabetic animals. We've had positive FDA pre-IND feedback that our GMP formulations are acceptable for an initial proof of principle clinical trial. They request only one additional animal toxicity study, for which we've been awarded $200,000 at the recent QED competition. We have a 505(b)(2) pathway for acceptance. We are seeking Invent Penn State funding for preparation of a proof of principle clinical trial, including the trial protocol and the IND application. We will then license our formulation. Diabetes is a global epidemic. Millions of people are involved and it costs billions of dollars. We will treat early stage diabetic wounds in order to prevent them from becoming late stage wounds and even resulting in amputations. There's a significant unmet medical need here. 14 to 20% of all diabetics experience a foot ulcer. Fortunately, about 70% of these ulcers are early stage. Unfortunately, over 60% recur. This is not surprising because the current treatment is routine wound care and offloading of the involved foot. Diabetic skin is different. There's delayed and incomplete wound healing caused by an overactive negative opioid growth regulatory system. When opioid growth factor binds to its receptor, it suppresses wound healing in many stages listed there on your right lower screen. Our research demonstrates that topical naltrexone directly blocks OGF effects, thereby normalizing wound healing. In the diagram you see in front of you, we are exhibiting our latest formulations, which are even more effective than our previous formulations and lead to even stronger wounds. Topical naltrexone is differentiated from its competitors because it alone goes to the basic pathobiology of poor diabetic wound healing. This is a huge global market for topical naltrexone, even if you only consider the early stage ulcers, which comprise about 69% of these ulcers. We have strong IP protection, which is owned currently by Penn State. Our business plan culminates in acquisition by a strategic partner. We will perform QED-funded swine safety studies. As required by the FDA, we would use Invent Penn State funding to prepare and submit our IND application and clinical protocol. We would raise funds, of course, for our clinical trial. Once completed, we would move to an exit. So therefore, with your support from Invent Penn State, we can alleviate this suffering of literally millions of people who have diabetic foot ulcers. Thank you.
Thank you, Joe. Very nice presentation. We will now start the Q&A with Alana. Alana, you can begin.
Thank you, Joe. Certainly a very interesting market. You mentioned a number of different types of exits, and I just wanted to sort of clarify and get my mind around it. You were talking about an exit within 24 months, and you talked about licensing, and you also talked about exiting to a strategic partner. So are you thinking of licensing to a strategic partner and retaining those fees that would come from that, those licensing revenues? Or are you going to just sell the IP? What are you thinking?
Well, obviously, if somebody comes to us tomorrow with a huge bag of money, we'll license it immediately. But anticipating that that will not occur, we'll probably license it to Elkos, who will be then charged with actual finding our final partner. Mark Lane, who is one of our...
That's not Elkos? Who's we?
Well, we being Penn State will license it to Elkos. Mark Lane, who would be the executive of that, has a considerable experience in bringing drugs to a market.
Got it. Thank you.
Sure.
Bruce, do you have any questions?
Yes. Thank you. Just trying to get off mute there. Great presentation, really interesting. So just to clarify, Elkos doesn't currently own the IP?
No, it belongs to Penn State.
I see. Okay. So a quick question about a clinical issue here. You know, diabetic foot ulcers are frequently coupled with peripheral arterial disease, you know, very prevalent in patients with diabetes. And a core problem here is, of course, restriction of blood flow to the foot or lower leg because of the arterial problem. So I'm just wondering, how would you untangle all that, given that the patient substrate you're dealing with is likely to have these arterial issues?
Yeah, I did 2 years of general surgery before going into ophthalmology, about a year of which was spent on vascular surgery. And as you say, the diabetics have two problems. They have large vessel atherosclerosis, but one of their major problems is small vessel disease. Our research has shown that naltrexone directly stimulates angiogenesis. And so if we get these early stage ulcers where we can still have viable tissue around the ulcer, we can stimulate blood vessels to grow in. And this should probably contribute to the lack of what we believe will be a lack of recurrence of these ulcers. So we obviously can't address the atherosclerosis, but we can address the small vessel disease.
Mm-hmm. And one last question. Obviously naltrexone is out there. You have some unique formulation. Can you just say a few words about how your IP addresses and protects that unique formulation?
Yeah, we have broad-based protection that it really includes all types of wound healing, epithelial wound healing, et cetera. So it includes multiple, or I should say all formulations of the drug. So the IP protection is so broadly based that it covers what we're talking about.
Okay, thank you.
Thanks, Joe. I have a question about the competition, and maybe you hit on it a little bit earlier when you were talking about the like the scientific merits of your product. But there's a lot that's being done in the space for wound healing. So, what exactly, in terms of like the functional output, is your product doing better than others?
Well, for instance, one of the drugs is only good for angiogenesis. So that's one of the 7 or 8 individual areas of wound healing that are attacked by diabetes. We address all of those. And other formulations do one or the other. They address fibroblast proliferation. One is an adenosine monophosphate stimulator, et cetera. But this goes in other research we've done in dry eyes, for instance. It also reverses dry eyes. So we found that the naltrexone path, or the OGF pathway, is fundamental in diabetes. And so this is the only drug out there that would address all of those different problems in diabetes.
But so for, I guess to clarify, for your specific, diabetic foot ulcers, since you're running animal studies, if you're doing like a critical defect model compared to other solutions in the space, are you actually seeing the wound heal faster?
Yes, and that picture that I showed earlier of the diabetic wounds, we had a control, and then we had, with our formulations, both the older and newer formulation, and the newest formulations heal the wounds much faster than either just an insulin-controlled diabetic or a diabetic that's given routine wound care, et cetera. An animal, that is.
Okay, great, thanks.
Sure.
Thank you, judges, for your questions. Joe, great presentation. Thank you.
All right. Bye, everybody.
Next presenters are Ali Demercy and Aidan Barajan of Vitamin K Fermentation. Ali, you may begin.
Good morning again. I'm Ali Demirci. It is my pleasure to talk about our invention on vitamin K production, and we call our company now Vitamin K Company. So vitamin K is a fat-soluble vitamin which has numerous health benefits. It might be interesting to know that vitamin K has 2 subtypes, K1 phyloquinone, which we get from green leaves, and K2 menaquinones that is produced by microbial fermentation, out of which menaquinone 7 is also known as MK7, is especially very effective in the prevention of cardiovascular disease and osteoporosis, which are the health issues affecting many people in the world and cost billions of dollars. Recently, MK7 is also shown to help recovery of COVID-19 patients. However, the major problem is that current MK7 production method used by the industry is very costly. The price of 1 kilogram of MK7 is $3.5 million. The reason for that is that producer microorganisms likes to form biofilm on the surfaces. Therefore, tray systems are used to grow them, which makes it not only difficult to control fermentation parameters, but also not able to scale up for large levels of production. Therefore, the million-dollar question is how to produce large quantities of MK7 economically. In our lab, we developed a biofilm reactor system called biofilm reactor using special solid support to address MK7 production problem. These supports are a mixture of plastic and microbial nutrients. We tie them on the rotating agitator in a grid-like fashion like this. During fermentation, biofilm is formed on these supports while we are still able to agitate the reactors. Just to summarize the outcome, this is how much MK7 produced during static fermentation like in trace, but it is not scalable and controllable as I mentioned earlier. When we agitate the system, the production drops to very low level like this. With our research using biofilm reactors and further optimization, now we are able to produce similar level of production in agitated systems, which can be now scaled up by using larger reactor sizes under agitated conditions. We have filed PCT application for our invention, and now we are working with the largest MK7 manufacturer in the world. Hopefully, we will be transferring this technology to the industry to produce MK7 at an affordable price for the patients. Thank you for your attention. Dr. Berengian and I will be happy to answer any question you might have.
Thank you, Ali. All right, we will now start our Q&A beginning with Alana. Alana?
Thank you. That was very, very interesting. And you mentioned that 1 kilogram costs $3.5 million, that's a lot of money. And I'm curious, using your methodology, maybe you don't have exact numbers, but I'd like some idea of how much it'll be reduced. By 50%, by 90%, what are you thinking?
Well, of course, it depends on the future market. What we think, the current technology is limited with the size of the trays that they can do. The tray cannot be as big as a football stadium. It is limited with the size, and also fermentation cannot be controlled on the trays like aeration and pH and etc. So in our systems, sky is the limit. We can make bigger reactors, like the largest reactors I've seen, 3 million liters. So we can basically make the product as much as needed by the market. So I cannot really predict what the future will be, but the company itself also very excited that this is opening doors for the great market in the future to help people at a affordable cost.
Thank you very much.
Yes, thank you. Very interesting presentation. Just to follow on that, how far have you gotten towards scaling this up, right? It seems like the key advantage of your process versus static fermentation is its scalability. So if 3 million gallons is sort of, the ultimate reactor, how far have you gotten?
Well, of course, as an engineer and scientist, we believe that this is scalable, but it is not, unless it is proven, we cannot convince the industry. Today, I mean, we have a project, again, funded partly by this MK7 manufacturer. I'm not able to disclose the name. And also College of Ag Sciences has a program, we call it RAIN grant, to translate the research into the commercialization. So with these two funding, currently, as we speak, we are doing a research in our pilot reactors to prove the scalability. So that's why Dr. Berengian with me today is working on that project as a postdoc, and he has been the co-PI since the beginning of this project.
And what's the end game for your company? Do you see yourselves licensing this off to manufacturers, selling the technology itself? What do you want to do?
Well, our end game is, of course, I'm a professor at university. I'm an academician. I am here to help the industry to make the production better and more cost effective. So hopefully, once we get our IP is approved, so we are going to provide the licensing to this company or maybe other companies in the industry to utilize this technology.
Okay, thank you. Alice?
Thanks, Ali. So we talked a little bit about cost, but besides cost, what are the other benefits of using your system?
Well, I'm gonna say that it is more importantly, the current technology is of course static and not scalable to large volumes. It's gonna make the production large volume. And also it is more importantly in fermentation, these are living systems. We have to provide the conditions for them. And in this agitated system, since reactor is more homogeneous when you agitate it, makes it more controllable. We can control those parameters to the finest point that there won't be any fluctuations in the production as opposed to other static systems. So if you produce this much, always you'll be producing this much because it will eliminate a lot of fluctuations in the production line.
All right, and I know you mentioned Aiden earlier, but can you maybe just talk a little bit about yourself and him and how you guys work together as a team?
Well, maybe I should let that Aiden to start because he has a longer history in this topic. So I added 5 years later than he started. Aiden?
Sure. Good morning, everyone. Thanks for having us for today's competition. Well, actually, it's interesting to mention, like I did my PhD in 2009 on Menarquinone 7. That was very early stages of Menarquinone 7. Then I knew Ali is doing research on biofilm bioreactors. And during my PhD, I found like these microorganisms produce biofilms. And when we agitate them, which we ideally want to agitate them because of the heat and mass transfer rates, and it's a better environment for cellular growth. So I touched base with Ali and I said like, hey, this is the issue we have. What do you think? Then in 2014, I became a faculty member in University of Waikato in New Zealand. And then we started this collaboration between both universities, which was amazing outcome, like lots of publications, IP, and now we have the biggest producer of MK7 interested in our technology. So yeah, that started from there and we built up on that.
All right, thanks.
Thank you, judges, for your questions. And thank you, Ali and Aiden, for your presentation.
You're welcome. And again, thank you for having us. This was a pleasure and honor for us. Thank you.
Thank you so much.
Our next presenter is Brad Rogers of Gradient T. Brad, you may begin.
Good afternoon. My name is Dr. Brad Rogers, and I'm the lead scientist at Gradient Technologies. Today, I'm very excited to introduce you to TempAssure, our lab-on-a-chip platform for measuring drug stability. Unstable drugs are a nightmare for the pharmaceutical industry because they're difficult to manufacture and they have short shelf lives. TempAssure's small-scale design allows scientists to predict these problems early on when their drug materials are limited. Did you know it takes over $6 million in 5 years to bring a single drug into clinical trials? The research phase of this process involves generating small quantities of many drug candidates and then selecting a handful that are potent and safe. Then a single drug is sent on to development to be produced in large quantities for clinical trials. In order for a pharmaceutical company to outrace their competitors to market, it's critical to avoid delays and failures. Choosing the right drug candidate, however, is particularly challenging because the amount of material is very limited for accurate risk assessments. TempAssure is a superior platform for assessing stability risks, which has the potential to reduce development failures by 25%. Our patent-pending design exposes a small amount of precious drug material to a range of temperatures simultaneously. And videos are recorded to watch the drug precipitate over time. The industry standard test for predicting stability only works for low concentration drug products. And our closest competitor offers an instrument that can't measure stability over time, and it requires by hand sample preparation, unlike TempAssure. Our business plan is a simple razor and razor blades model. We offer a $100,000 benchtop instrument that uses $50 consumable sample chips. There's a significant need for this product in the biopharmaceutical space, where we expect $100 million in instrument sales and $40 million every year from the consumable chips. In fact, we've already raised over $1 million through collaborations with AstraZeneca and Sanofi to test this platform on their drugs. My team at Penn State and our biopharmaceutical collaborators are currently focused on prototype development. We plan to form a company and begin manufacturing in 2022. And by 2023, we will launch sales to dozens of biotechs across the world, thereby eliminating stability risks and accelerating times to market. Why settle for being sure that a product is stable when you can be temperature sure? Thank you for your time, and I'd be happy to take any questions.
Thank you, Brad. Nice presentation. We will now start with the Q&A, beginning with Alana. Alana, do you want to kick us off?
Sure, thank you. That was a great presentation, very interesting problem you're solving. Tell me a little bit about the manufacturing side. You mentioned that you need to form a manufacturing capability. Have you done some studies to determine that this is indeed manufacturable at scale, reliably, and at the price that you're quoting, $100,000 cost? And that includes those sample chips as well.
Yeah, that's a great question. We're early on, so we're actually still developing the prototype, and we have the raw materials are about $10,000. So we're going to have to explore these questions you're mentioning. How does that actually translate to large scale manufacturing? And we're actually very interested in partnering with some existing instrument manufacturers to hopefully eliminate some of the costs on our side and leverage their existing relationships in this space.
Mm-hmm. Thank you.
Thank you for the question.
Brad, very nice presentation, and congratulations on raising $1 million already. That's quite an achievement at this stage of the game.
Thanks.
I'm a little bit curious about what did you do, or what are you doing with that cash, and can you speak a little bit about the results you've seen with whatever compounds AZ and Sanofi have sent you?
Yeah, so we've collaborated with these pharmaceutical companies for about 5 years now. The first projects with AZ were focused on formulation chemistry, and we published a paper in 2019 with them. And over time, these projects kind of evolved to develop our instrumentation for predicting shelf life. And I'm really excited to share this, but last year in our collaboration with Sanofi, we did a blind study where they provided us with a handful of their drugs, and they know the shelf life, and they asked, can our instrument predict that? And we got some really promising results that shows there's a strong correlation, and we're writing that up into a manuscript that will serve as our first major piece of advertisement.
How did they react to your result?
I think very, very excited. We actually had a meeting with them a month or two ago talking about potentially onboarding or taking our prototype for a test drive in their lab. That's what they see as the next step. They want to try it out for themselves.
Okay, thank you.
You're welcome. Thank you.
Hey, Brad, thanks for the presentation. So does your device only cover small molecules, or can you work with other like biologics, for example?
Yeah, good question. We've almost primarily focused on biologics. Monoclonal antibodies are the biggest biologic on the market right now, and that's what we've been focused on. It's an open question as to how big of a scope can this technology be applied to, but we've primarily focused on biologics up to this point.
Great. What's the temperature range that you can, I guess, your gradient covers, and what's the sensitivity if you're able to fine tune that?
Yeah, our hardware can go anywhere from -20 on the cold side, that's degrees Celsius, up to the boiling point of water, 100 degrees. So that's plenty of room for a biologic, but for other materials, we might have to explore different designs to go farther. As far as fine tuning goes, you're limited by sort of the microscope on spatially resolving these temperature gradients.
All right, thanks.
Thank you.
Thank you, judges, for your questions, and thank you, Brad, for your presentation.
Thank you for the opportunity.
Our next presenter is Catherine Lawson of Hydrosapient. Catherine, you can begin your presentation.
Thank you. Hi, everybody. I'm Catherine Lawson, CEO of Hydrosapient, an academic technology transfer startup working in water resources. We have multiple projects all centered around our mission to accelerate global accessibility to reliable and timely water resources predictions. Current stream flow predictions are inaccurate. It's hard enough to predict the weather, and it's unfortunately not as simple as more rain causing more stream flow. Flooding we can predict is already bad enough, but flooding outside of predictions results in billions of dollars of inland uninsured water damage and causes homelessness and bankruptcies. Available flood maps are notoriously inaccurate, and the rapidly growing private flood insurance market is demanding better information. We're developing solutions to tackle this problem and are approaching stakeholders in that market. Beyond flooding, stream flow volume predictions are extremely valuable for irrigation, reservoir management, aquaculture, wildfire prevention, and more. Hydroelectric power generation alone is a $60 billion market, which is actively seeking better stream flow predictions. These predictions are also useful for hydropower users like cloud services and cryptocurrency mining, and can further incentivize their use of renewable energy. We believe Hydrosapient can revolutionize these predictions. Hydrosapient's chief differentiating factor is the continuous facilitated infusion of new science from our Chief Science Officer's research group at Penn State. Dr. Xiaopeng Shen introduced the concept of applying deep learning, an advanced form of artificial intelligence, to the field of hydrology. And his research group is leading the field of hydrological modeling. I am Hydrosapient's CEO and have a decade of experience building and leading engineering and research teams. I'm an environmental engineer with graduate degrees from Penn State and Ohio State. We're advised by Jan Dutton, CEO of Prescient Weather, who has over 20 years of experience with weather forecasting-based products. Today's commonly used models for operational flood forecasting are only 64% accurate. Hydrosapient's median accuracy across the entire continental United States is 86%. It's a 34% improvement already. For hydropower producers, this could be worth $4.1 billion a year. This unprecedented performance is possible through a novel combination of deep learning with an understanding of hydrological principles. And our team has more novel developments showing promise. Our short-term roadmap shows a few products nearing launch, all of which are highly scalable and have high profit margins with a business-to-business model and online subscription-based platform. Future products may include a method to improve resolution for satellite measured variables, which has been independently noted as a problem in our customer discovery interviews, as well as a method for improving existing physics-based models, meaning better prediction of environmental factors that we can't measure with satellites. So what would it mean for us to win the tech tournament? Money from the tech tournament will allow us to expand our team, so we have the person power to convert the research code into a more robust, user-friendly web-based platform, our minimum viable product. This would enable us to approach potential customers with a working product in hand within months and allow us to fund our own development. I appreciate all of you attending and listening, and I'm happy to answer any questions you may have. Thank you.
Thank you, Catherine. Nice presentation. We will now start the Q&A, beginning with Alana. Alana?
Hi, very interesting, Catherine. Thank you for that presentation. Can you tell me a little bit about the customer discovery you've done? You have this wide range of applications and you talk about a B2B model, and I'm just curious, what kinds of customers have you talked to and what have you shown them so far that's helped you to choose which of these markets go first and how to make money on them?
So that's actually a big problem. One of our big questions as well is which markets and where, because there's so many applications of this. And we're still currently in that process. I presented a few of the potential markets, but we have been in discussion with potential customers. I don't know if I can name company names or anything, but I guess significant involvement in the weather forecasting industry and others.
Okay.
Yeah, sorry.
Thank you.
Very interesting, Catherine. Let's talk a little bit about the business model that you envision. It sounds like you're going to put this up on the web and extract value from the people who are using it somehow. Can you tell us about that?
Yeah. So what some of these companies that would be using our predictions would do would be that we would tie in they would tie in their interface to our interface. So they would basically send us a ping or like a message. I'm not a software engineer, but basically their system would ask our system for the most up-to-date predictions, and we would send it to them, and then their user interface would display.
And you're going to charge them per query or something like that? Is that the plan?
Yeah.
Okay. One other quick question. How will you protect this invention or inventions?
Well, for some of the inventions, we're proceeding with patents. We have a draft patent application right now that we're going through with lawyers. For others, it's not so much protecting the intellectual property itself as it is continuing to move forward. And so that's why our push is really on moving the field forward and continuing to integrate developments that are just coming out, that are just becoming aware, because we don't want to close off research that can help people. You know, we don't want to ask Dr. Shen's group to not share their developments. And so by continuing to move, we think that's the best combination of humanitarian work as well as company involvement.
Okay, thank you.
Thank you.
Hey, Catherine. Hey, thanks for the presentation. How much are your customers paying today for this type of data? Like, do they have a budget?
So depending on the customer, so some would have budgets, yes.
Okay, is it like a substantial amount? Is it something that they, I guess, is integral to their decision making, or sometimes they don't have it? Like, it, you know, I don't know what data sets are out there today.
Okay, yeah. So some customers would have a budget for obtaining the best predictions, and one of the, I guess one of the weather forecasting companies that we have talked with, they basically said, you know, we want the best predictions. If you have the best prediction, we're going to be interested in being a customer. And that was essentially the,
Okay.
I guess the message there. There's others who, for example, in the irrigation industry, who may not be actively seeking solutions, and so that would be more of developing a new market. Whereas for hydropower, for example, the companies are already seeking. There's actually a couple competitions ongoing from an industry group, and they send out a couple of requests for proposals to get a current status report of predictions.
Awesome, thank you.
Thank you, judges, for your questions, and thank you, Catherine, for your presentation. Our next presenter is Nate Watson of X-Form Technologies. Nate, you may begin.
So hello, my name is Nate Watson, and I want to introduce you to X-Form Technologies. You're probably familiar with plastic desktop 3D printers, but X-Form Technologies steps this concept up with concrete 3D printing. Similar to plastic printers, concrete printing uses concrete extruded through a nozzle to form a 3D shape layer by layer without the use of labor-intensive formwork. Using this technology, we intend on being part of the solution of a problem spread across the entire construction industry. Labor issues and shortages within construction, driven by the lack of productivity, has placed it in a position ripe for innovation. As time has gone by, the public perception of construction is that it is dangerous, difficult, and dirty. As industries such as agriculture and manufacturing have invested in technology and innovation, the construction industry has been relatively stagnant. We have found by looking at precast concrete products and visiting facilities, and by talking to precast concrete manufacturers, that they would be a great first customer segment to pursue. Precast concrete manufacturers have facility conditions similar to the lab and produce products at the size that our system has produced. The precast industry is also quite large, with 1,827 US companies and a market size of over $130 billion. What we have heard from precast companies is along the same lines of what we see with the stagnant construction industry as a whole. Labor is a limiting factor and prevents them from completing all their potential bids. They lose money on custom designs in order to maintain high-value customers. And they experience high job turnover costs. And if they want to make a new product, the formwork for it can be very expensive with long lead times. Our business seeks to alleviate those pain points using our technology by increasing per worker productivity, removing the need for custom form work, changing the job perception through automation, and allowing businesses to diversify products without the cost of new forms. We plan on doing this while also creating a new business possibilities for our customers as a side effect, because as we know, to enter any market, especially a conservative one, being able to solve their current problems costing them money now is going to get us in the door. And being able to improve their products and help them attract a broader group of happy customers will get us in many more doors. This differs from our concrete printing competitors who want to be their own construction companies and compete with current construction practices as a whole. Our strategy is to work with construction companies by addressing their pain points and grow our market segment with the technology rather than trying to compete with the entire industry as a whole. After developing our award-winning system at Penn State, which won nearly $500,000 in competition winnings, and our award-winning participation in the Tech Accelerator and the completion of the Summer Founders Program, my co-founders and I are ready to use our expertise in additive manufacturing and design, mechanical engineering, systems engineering, architectural engineering, and architecture to make Xform's technology a success. Xform technology is now ready to continue development with close contact with excited potential customers, begin consulting with interested universities, pursue SBIR grant funding, and work alongside Penn State for testing to make sure our product is as good as we think it can be. Thank you so much for listening, and I look forward to answering any of your questions.
Thank you, Nate. That was a nice presentation. All right, we will now start our Q&A, beginning with Alana.
Thank you, Nate. That was great. I have a question. You talk about precast as the main competitor, and I imagine that it's much cheaper and, you know, better suited, let's say, to making hundreds or thousands of the same form again and again, and that you're better for individual to custom. Is that correct?
So yeah, so the precast industry suffers from limited labor, which when we talk to them, they're addressing their current pain points now, which is the fact that they don't have enough labor, and the labor is getting because of shortages and that limited supply, the price is also increasing, and the turnover cost from hiring and losing people is also a driving factor. And so what we claim is we can come in and help increase that per worker productivity by eliminating the need for the multiple workers needed to produce, particular products. And then as a side effect, basically our technology will also provide zero cost extra for custom designs, as well as allowing businesses to diversify their products.
Okay, so I get why on a custom design that would make sense. But I'm wondering what % of the market is actually those custom designs? And I guess if you could explain to me, why would this be more cost effective than precast in a situation where you're making the same thing again and again, hundreds of times?
Yeah, that's a great question, because, so you would think that precast is the cheapest way of producing the same product over and over again. Mm-hmm. And that makes sense, but what we hear is that it takes, because of the especially the reinforcing process, the molding and then demolding process, it ends up taking many workers to produce one part, where using our technology, it may take one worker to operate the machine, and it can continue to produce concrete products with a lot shorter turnover because of the curing process, but also just fewer workers as a whole, just because it doesn't require, sometimes it might require 4 workers, hours of work just to do one form, even though that they own the formwork. Yeah.
And so once the 4 workers do it, they can't make 1,000 pieces each time it takes 4 workers?
Yes, it does.
Oh, wow. Okay.
Yeah, it can cost thousands of dollars in labor costs to create some of these forms.
Okay, thanks.
Nate, very interesting. Have you gone as far as to sort of sit down with one of these customers or prospective customers and figure out, how much you can reduce their cost of goods on a routine piece? Can you cut it by 20%, 50%, 70%? What is it?
So we have talked with multiple precast manufacturers and kind of looked at their product portfolio and to see what the technology can do now versus what requires development. But in terms of the actual cost cutting per product, I think that's harder to gauge at the moment. Although part of our targeting is to create like the productivity of our system equivalent to like man hours or a person hired, for example, and create our leasing price structure cheaper than what our system can produce, basically.
Your business model is to lease the equipment?
So it would be to sell the equipment, and there are our hypothetical sale would we're thinking might go through a leasing company or a bank or some sort of form, but we would probably just sell the company, just sell the product. Sorry.
Okay, thank you.
I know I'm supposed to ask a question, but I just want to follow up on that. Robotics as a service is also really hot right now. So it's a form of leasing, but you can subsidize the cost of the equipment, and they would pay per, I don't know, square foot or whatever that they would be manufacturing. But that leads me into my question. So I'm thinking about scale. So size scale and time scale. So in terms of size, these concrete can be really large, especially if you're using them for building. So how large is your system capable of handling right now? What dimensions, and what percentage of the market does that cover? Yeah, so that's another point about precast concrete is often the size of the products that they're producing. It needs to fit on a flatbed truck, and so that's part of the reason why I think a lot of the precast products are actually can be smaller. I mean, obviously it can be much larger with wide loads and things like that. But in terms of size, you can imagine a flatbed truck and what can fit on there. I know from the last slide you could see part of what we had printed before in competition as being roughly that size of maybe a half of a flatbed. But we're excited to continue development and scale that up with likely moving the robot and things like that. But it ends up being a very large market size. Yeah. Awesome. One last one on the time scale. You talked about man hours, but also the precast takes a couple of people. But how long, I guess, end to end linearly does your process take versus the conventional method? Yeah, so the printer needs to be obviously operating the whole time versus letting pouring and letting it sit. But if you think about it as like because of the manufacturing type with 3D printing, it requires the material sets, and so basically right when the print is finished, within probably around of 30 minutes, 0 to 30 minutes, the concrete part can be moved off of the manufacturing floor, which opens up more space and can help with the turnover for the machine to print another product versus having a bunch of forms, filling those forms with concrete, and then having to wait for that concrete to cure enough to demold it before you can reuse that form. Thanks.
Thank you, judges, for your questions. Those were great. And thank you, Nate, for your presentation. A very interesting market.
Yeah, thank you.
Our next presenter, Barry Fell and Randy Howlett, representing Cranial Devices. Take it away.
Thank you, everyone. I'm going to introduce HydraFix to you, a minimally invasive surgical solution for hydrocephalus. Along with my co-founder, Dr. Randy Howlett from the College of Medicine, we were the previous first place winners in the 2016 tech tournament, co-founders of SIG Medical, which was later sold to Zimmer Biomet in 2019. Hydrocephalus is a $3 billion annual healthcare cost. Roughly 1 million patients are currently diagnosed with the disease in the US. Hydrocephalus is too much CSF fluid around the brain. There is no cure. Patients undergo a lifetime of repeated surgical treatments, and hydrocephalus shunts have the highest failure rate of any medical implant being sold today. The initial market segment that we're going to focus on is NPH, normal pressure hydrocephalus. It's primarily found in the elderly. Symptoms are often confused with Alzheimer's, but the symptoms can be reversed with shunting. Currently, there's 750,000 patients with NPH diagnosis in the US. However, only 1% of the patients are currently being treated because of this high failure rate of the implant. The conventional shunt problem is that they're too long. They siphon and they overdrain the CSF. Overdrainage in the NPH patient can lead to fatal cranial bleeding. NPH patients deserve a better solution. For the right product, a significant market opportunity exists. By 2025, there'll be almost 1 million NPH patients in the US. If you serviced only 1% of that population, that market opportunity is almost $70 million. The HydroFix solution is elegant. HydroFix will drain CSF directly into the venous system. It restores the normal physiologic pathway. It significantly shortens the shunt length. It eliminates siphoning and overdrainage problems. It's the perfect solution for NPH patients. Cranial Devices was formed in November 2020 to bring HydroFix to the market. We already have an experienced leadership team in place, strong IP, comprehensive development plan is already in place. HydroFix will be on the market in the first quarter of 2023. Patients, surgeons, hospital payers, they'll all benefit from its use, resulting in a rapid market adoption. In summary, it's a large, distinct US market opportunity with worldwide appeal, strong IP position. HydroFix will be able to gain a breakthrough device designation under the new CMS policy called Medicare Coverage of Innovative Technology. We'll be seeking roughly $7 million in financing through 2022. We expect to be profitable by 2024. We already have high interest from major medical device companies and private equity. Thank you. Okay.
Hi. So I see that you were, one of the things that you were thinking about talking about was your use of funds. So yes, perhaps you could tell us a little bit about how you're going to use the funds.
Sure. You can, I'm sorry, everything's going through too quickly at this end. Okay, that's our timeline. If we're going to do a 510K pathway, we could be on the market by January 2023. However, we realize that there's a benefit to doing de novo or PMA because of special controls. There's actually more value to the investors if we go that route, which would push our market release out into 2024, 2025. Okay, so if we're going to do just 510K, then we estimate that we will raise about $12 million to do that for the 510K pathway. Obviously, it would be more if we do anything involving clinical trials.
And have you made a determination which way you're going to go?
No, we've got a pre-sub meeting coming up here in a few months. Okay. We'll probably have several meetings with the FDA. But it's reasonable to assume that we'll have either a very limited market release if we do go 510K, but most likely we expect to go de novo or PMA. All right.
Very interesting. Thanks for the presentation. A couple of quick questions. One is, what sort of chronic animal data do you have that establish that this, in fact, will last much longer than the current device?
Well, sheep is the animal of choice for these. But also, there's about 30 years of experience out there already doing this type of drainage into a sinus. Just right now, they're just taking a simple catheter to do it. And the failure rate of that is extremely low. So we know it works. What we need to do is make it a more standardized procedure.
Okay. One other quick question. You guys have been at this for a while, and nobody knows better than me that it takes a long time to develop these kinds of things. But I'm just curious if any of the big players in the space have come knocking, and if you've talked with them, what has prevented them from moving on the company already?
Nothing, but we've been trying to keep kind of low profile, but yes, we've already had a few pretty hard knocks on the door.
What was their reaction?
Follow-on meetings. And but again, we are somewhat limited on how much we're willing to disclose, but also just I didn't get to that the IP slide, but we had some provisionals that were converting here. In fact, we'll convert this week actually, and they won't be published for another 6 to 8 months probably.
Okay, thank you.
Sure.
Thanks, Barry. I have a question about the strategy. So you talked about the 1 million hydrocephalus patients, and you were targeting the majority of those patients. Did you ever consider targeting a smaller, like a rare disease set of patients? Would that make sense at all?
Yes, I mean, the one that tears your heart out is the pediatric market, but that's actually a more complicated one. The forms and causes of hydrocephalus are a little bit different there. Plus you have to deal with the growth of the child. The adult market, especially the NPH, you have I mean, not to sound, kind of cold, but you have a finite amount of years you're going to live. And if we can improve the quality of life, we can really produce enough funding to then go after the pediatric market.
Okay, thanks.
Okay. Probably more key here is, I didn't get to it, but last week the CMS policy was changed. So the MCIT program that they started guarantees funding, or essentially payment of any device for under Medicare for 4 years after your release of the product. That's significant, because now we're not fighting which codes are we going to use, even though we probably think that we meet the existing codes that are out there now. This kind of clarifies that point completely.
Can I ask Randy a quick question? I'm curious, how much more difficult would this be to implant than the standard version?
That's where the challenge is. So as you saw, in that slide, we want to put this into a dural venous sinus, and that's where the challenge and that's where the opportunity also is. So our IP is centered around localizing and precisely placing a very low profile outlet or intrusion into that dural venous sinus. So there is a little bit of a hurdle there. But again, that's where our IP is, and again, that's where our opportunity is.
Thanks.
We have a pretty substantial fence around us from an IP point of view.
As Barry said, this approach, shunting directly into this dural venous sinus, is known to work. There are challenges, as you said, Bruce, and then there's a lack of standardization in the procedure.
All right, thank you. And we are at time. So judges, thank you for your great questions. And Barry and Randy, thank you for an excellent presentation.
Wow, what an awesome group of technologies today. I want to thank all the presenters for their wonderful presentations and introductions to their company. I'd also like to thank Erica and all of the judges, Dr. Alice Chang, Alana Diamond, and Bruce Shook. I think we can all agree that the judges did an outstanding job quickly assessing each company and asking thoughtful questions. We also appreciate their time and expertise. We will now leave our judges to the hard task of deciding on the winners. The winners of this competition will be selected by our panel of judges and will be announced tomorrow, directly following our keynote speaker, Kenneth Fraser, Chairman of the Board and CEO of Merck & Company, Incorporated. Kenneth will begin speaking at 2 p.m. Again, I'd like to thank our 22 generous sponsors for their support. Up next, please join us at 4:25 for session number 3, Life Science Ventures Addressing COVID-19. There's a lot going on, so please refer to your program book or the website for the complete schedule and details. Thank you for attending.